Module 4, competition. So before I get started, I want to just take a little step back here and explain to you what the thoughts were behind this, as David and I thought about how to present this and have you guys interact. So this is really, as David mentioned, where the rubber does hit the road. These are the people that some of your customers will ask you about, and there's many of them. There's some really big institutes out there, but if you go online and check them out, it's not always obvious what they can do. They are clearly doing cancer, but they may not necessarily be doing next generation sequencing, okay? So that's a big thing to bear in mind when customers who don't quite understand what it is you're doing and what these places do. So you're doing next gen sequencing, and that's really it. Some of these other places do more than that. They do next-gen sequencing, they do FISH. We talked about that yesterday, fluorescence in situ hybridization. They'll do PCR. They'll do all sorts of interesting ways of getting to understanding what that patient material is, whether it's got cancer in it or not. It may just be simply looking at the slither of tissue that is taken from the patient. That's it, just a pathologist. There's a lot of things out there. So focus mainly on the competition with respect to next-gen sequencing, okay? It was pretty tough in some places to actually find out even what platforms they were using, but they were touting next-gen sequencing. So we'll go through it. I've split it up into two parts. One part is really the hospitals, the academic environments, where all that research gets funneled into that CLIA lab that I was talking about. That's where all the regulatory work is done. And then there's the private companies like yourself, and I think I have one public company, which is of course our friends at GE Healthcare, Clarion. Okay, so hospitals that provide the genetic tests. First one off the bat is the Baylor College of Medicine. This again is a big institute down there in Texas, and they have a medical genetics laboratory. So these guys have always been very prominent in the field of medical genetics. If you go to ASHG, they're there. People have gone into that environment and come back out, have set up their own businesses or gone into academia. So for them then, and this is interesting, this cancer gene mutation panel seems to be a common panel that is out there. It has 46 genes in it with 739 mutations. It seems like some of these academic / CLIA lab type environments are using the very same panel. And you'll see that come up in a few places. In this case then, they're looking at a broad selection of cancers, everything from all the stuff that you folks are actually dealing with. There's a few other extra ones like the MPD and the AML. They're using the iron tolerant PGM. They take all these samples, blood, bone marrow, FFPE, and fresh frozen tumor, but their turnaround time is 14 days. Okay? And then at the bottom of each one of these, I've put their website that you can go to, so just click, go straight in there, and delve a little bit deeper into understanding what each one of these people do, or each one of these sites do, and how they do it. But this is interesting, as I mentioned. This cancer panel seems to be quite a common panel that is used by many of these type of institutes. And so you'll see it come up several times. Okay, we got University of Washington up in Seattle. They have their Department of Laboratory Medicine. They have several products out there. They use the Oncoplex cancer gene panel, the Broca, and the ColoSeq. This one here, the Oncoplex, has got 194 cancer-related genes on there, and they also look at gene fusions and rearrangements. The Broca cancer risk panel has got 40 genes on it, and then the ColoSeq is more around Lynch and polyposis, sort of a hereditary type, and they have 13 genes on there. They use the Illumina instead of the PGM, the iron tolerant, and they have a broad range of cancers that they're looking at there. So same type of samples that can be sent in, the FFPEs, purified DNA. Again, some of these sites might, some of these places may have the ability to take purified DNA, peripheral blood, as well as bone marrow. Their turnaround time is 8 to 12 weeks. So big difference, big stark difference then between 14 days here at Baylor College of Medicine versus UWASH coming in at 8 to 12 weeks for some of these panels. So again, as I mentioned yesterday when I went through the competition, you guys are standing strong. And if you created a nice table with some of these key parameters in here, looked at, as you've seen them, sort of like a circle which has got 3/4 filled out or completely filled out, or 0, not just blank, see how you compare. I think you find that in some of these key areas here, you're definitely full black circle there, showing that there's some really good, strong positive benefits. 8 to 12 weeks is not a very good turnaround time when you're sitting there wondering what's going to happen next and what's the best therapy. UPIT was a good one that I found. They have the same panel that I was talking to you about, that cancer panel with the 46 genes on, but note how they have a different range of cancers that they're using it to identify the molecular signature of some of these diseases. Clearly breast, colorectal, lung, obviously a common one and a clear one, but it's got the brain cancer on there, the ovarian cancer they're looking at, as well as renal, and the melanoma. They too use the PGM, and they only take these type of samples, either a fresh frozen tumour or the FFPEs. Now they got a faster turnaround time here, 7 to 14 days, compared to the previous guys, UWASH, which was 8 to 12 weeks. So that's kind of interesting, and they're in keeping with the folks at Baylor College. So obviously there's a group of guys that got together and said, hey, here's a set of genes that we can use, put it into our clear labs. There may be a relationship between them that's going on. I wasn't clear what the connections were, but that 46 gene panel with those number of mutations in there is clearly something they do quickly and effectively over a short period of time to get you answers around some of these diseases. However, the diseases are different. So MD Anderson, that's another big place, and first thing your eyes go straight to is the cancer panel. Again, they're sharing that same cancer panel in there, and they too have a different set of uses for it. Not only are they looking at human malignancies, but they're also using it for a prognostic and therapeutic applications, which is interesting and something that is of great value when you're looking at molecular signatures. We talked about it yesterday. It tells you not only what therapeutic treatment you can go on, but it could also tell you what your relapse could look like too. And so when you're looking at prognostics and therapeutic applications, you start to delve into companion diagnostics, which is big. And there's going to be, I'll have a couple of slides around companion diagnostics to help you understand that term and the value that clinicians are looking at when trying to determine how to utilize these molecular signatures for their patients in regards to the therapeutic treatment. So these guys are taking just simply whole blood, which is good for patients. That's nice not to be totally invaded, as well as some of the FFPEs, which of course naturally means you have to have some kind of tissue resected out from various parts of your anatomy, depending where the cancer is residing. The turnaround time here is getting close to you guys here, 5 days. So these folks are really pumping out some data using this 46 gene panel. And then a couple of other places that were hospitals where I did try to dig in. These are big places, and we talked about them yesterday. Some people were talking about the Sloan Kettering and the Mayo Clinic. It was very difficult for me to find out what they were basically using from a next-gen sequencing panel and what was on it. But they clearly are saying that they're using next-gen sequencing. They're clearly saying they have some kind of cancer panel, but it's not quite clear whether it's in a clear lab, whether it's still in the research part of their wing, and they're moving it in there. But these are big guys out there that people know about, and you will probably get asked, well, doesn't the Mayo Clinic do that, or doesn't Sloan Kettering? I would recommend that you take a little time to dig in there, or if you've got some intel out there that you can touch on and sort of understand what's going on. That's why good meetings like the American Society of Human Genetics is good to go to, because these guys will be there. They usually have their stalls, their stands in the big halls, and you can go out to them and talk to them a little bit about what they're doing in that, especially in respect to the use of next-gen sequencing. But it wasn't clear to me that they've actually launched into it in any big way. So just bear in mind, though, these guys probably aren't too far behind. Okay, so going to those private businesses, much like yourself, who's doing what? A surgeon, I found these guys have a panel that's particularly focused around colorectal cancer. They're using the ion torrent. They're just looking at basically two genes, KRAS and BRAF, and we talked about those yesterday and their implications from a predictive standpoint. They just literally take the FFPE and within 8 to 10 days you will see that the results come back, which is again not as good as you guys, but it's still a competitor with regards to that turnaround time. Foundation Medicine, they have their product called Foundation One, a sort of what I call a pan-cancer test. It's just basically anything that comes in, put it onto this panel here, which is 236 genes, 3,769 exons and 47 introns of 19 genes involved in rearrangements. I'm out of breath just reading that. And by the way, these aren't the only people that are doing that. Some of the sites that I went to also were asking, hey, if you are comfortable, try to remove the patient's name, de-identify it, send us the data, let's collect this information and see how far we can get, because everybody's doing this in their sort of independent locations. How can we bring it together into a database? But again, it's not centralized. It's still somebody's trying to say, send it to me, send it to me. So that obviously they're looking for those connections so that they can create products that they can provide to customers. So it's kind of an interesting way of sort of saying, hey, we're helping you if you can send us all this data, but you wonder at times what's behind it. Molecular MD is an interesting company that sprung up. It's a small little company, but they're definitely moving ahead in trying to get to providing a service to customers that looks at some of the mutations in just a handful of diseases. We talked about the GIST, the melanoma and lung cancer. And they're using the AmpliSeq and TruSeq cancer panels. It's not like they've actually designed their own panels. They're just taking what's off the shelf, putting in the clear environment, and using these two platforms to utilize the AmpliSeq and the TruSeq. The TruSeq goes on the Illumina platform and the AmpliSeq goes on the ion torrent. And of course, both those panels contain multiple genes. And it wasn't clear from when I was looking around on this web page what their turnaround time is. But they do take just FFPE samples. Okay, I believe David, I think David alluded to a press release just recently. I actually saw that press release too, and to me, when I read that, it was like, yeah, you know, we're in there. It's like notifying all and sundry, we're in the game. But when they'll actually have this cancer panel ready to go is going to be something that you folks, as you do your intel, will need to realize. Now, bear in mind that Clarient Healthcare bought Clarient, but they also bought, oh blast, a sequencing lab, SeqRite, down in Texas. And they are, so between Clarient and the sequencing lab, check out SeqRite, SeqWright. They have all that technology. They do both platforms, Illumina and the PGM. They also do a lot of the other sequencing or technologies as well. So Sanger sequencing still occurs, but with the fluorescence that I talked to you about yesterday. So between SeqRite, Clarient, and then GE Healthcare holding them together, there's going to be some development and design, and then it will be put into clearly into the Clear lab, which is the Clarient lab. And the guy that owned that company now works, Ronnie Andrews now works at Life Technologies as the divisional president there, bringing all their business together. So it's a very interesting environment. Small, but they all move around. So again, these guys, it's not clear what their turnaround time is. Again, it's more, I guess, a press release just to notify, put everybody on notice that they're coming into the game. And this is what they're going to be putting out there: tumor suppressor genes and oncogenes, the most common ones to be used on FFPEs. Moving to Ambry Genetics. Now, Ambry Genetics was an interesting company because they're sort of tied into some of the technologies that I talked about. Raindance Technologies is a company that makes the makes the little emulsion beads as part of the process towards sequencing, and they have their instrument in with Ambry Genetics, and they use next-gen sequencing. However, it wasn't clear what platform they were using, but it was clear that they were using next-gen sequencing. One of the interesting standouts here was that while they have these nice products, their hereditary cancer panels, they don't seem to want to bring the data to the customer in any expeditious manner. So 12 to 16 weeks is quite a long time, but you know, they probably think that there's no need for that from a business standpoint because it's more of a hereditary. But still, if you are expressing the cancer phenotype, then irrespective, to know whether you have a cancer mutation or a hereditary one that's driving it is important from a great, now we can put you on this therapeutic treatment. So anyway, these guys, it's 12 to 16 weeks, blood or saliva, and they have a breast cancer, colon and ovarian cancer panels with a various assortment of genes on there. I can't remember if these guys revealed what the genes were, but again, you have the link and you can check them out. Correlagen Diagnostics, I believe David sent out a pretty nice slide deck to everybody that alluded to what they get up to. Again, these folks are into the hereditary form of colon cancer, as we've talked about, Lynch's syndrome. They use a sequencing platform, but what type, it was unclear. They just have 4 genes in various multiple combinations. So you can pick 1 gene, you can pick 2 genes, you can pick 3 or all 4 in those various combinations. They take whole blood, and it wasn't clear what the turnaround time was, but they made it quite clear that it was short, whatever that means. Now GeneWiz was interesting because I know this company from the fact that if you want to have anything sequenced, you just send it to them. They're a bit like SeqRite. SeqRite and GeneWiz are sort of competitors. You literally send your DNA samples to them. It can be, we talked about yesterday, putting your DNA into a vector called a plasmid, if you're doing some kind of science work. If you want to have anything sequenced, you just give them the product, and within 24 hours, the data is sent back to you via email, and then you can start to work with that sequence. You can just check to see if it's 100% perfect, and then you move on with the experiments or whatever. But GeneWiz has moved a little bit more into research and has a CLIA lab, and they have a clinical sequencing panel which for EGF and KRAS. Now they've also developing a couple of other panels which they touted, which aren't ready yet, but the one at the top there, the EGFR and the KRAS one is, and it's for the non-small cell lung cancer and colorectal cancer, as well as other major cancers that we see EGF and KRAS. You saw yesterday when I went through each one of your panels, those 2 players are key in many of the pathways. So for them, GeneWiz is sort of saying, well, let's get started on this business model and just get working on these 2. They do have technologies that allow them to use any platform that you like, and you have the opportunity to say, oh, I like the Iron Torrent or the Illumina one. This cancer panel then has 4 genes in it on top of the EGF and the KRAS, but again, they come in various combinations, and they take all sorts of samples. Now, their turnaround time for research is typically 24 hours, so I'm kind of interested from a clinical standpoint how quickly they can do that too. They didn't say. But they're very much early in the game here. I wouldn't consider them too much of a threat just yet, but they're definitely starting to step up and become a potential player because they have everything ready and they've been around from a research standpoint, just like SeqRite. So GeneDx is another company that's working just on hereditary cancer syndromes. They are touting 27 of these types of syndromes. They use the ABI 3500DX. This has just got in February FDA clearance, 510K clearance as an instrument. Remember we talked about instruments need 510K clearance, and remember that Illumina as well as Iron Torrent have submitted their pre-IDE to begin the process of 510K clearance for their instruments, the MySeq and the PGM. So Life Technologies just recently got their 3500DX, and it's a capillary sequencing which basically means that just like I showed you yesterday with all those nice colors that you saw lined up, it just goes down a very, very thin capillary, and there's a sort of monitor that measures all the colors as it comes by it, but because it's so thin, it gets done with small sample amounts and keeps the cost down. Myriad Genetics, I threw them in there, not that you guys need to know about these folks. They're front and center when it comes to everything breast diseases, especially with regards to BRCA1 and 2. They're always in the newspaper with regards to the patents, should they own the mutations and so forth, and they seem to have managed to have kept the patents on their side while they're still arguing about that whole role of the mutations and ownership. It's something that is out there. These guys do have a turnaround time of 2 weeks though. Again, they're just looking at these hereditary cancers for a broad range. You think only breast and ovarian, but they have migrated into colon, uterine, melanoma, as well as pancreatic and prostate. I couldn't find out what instrument they are using. It wasn't quite clear from their website, but their panels tend to either be single or multiple genes associated with those diseases up there, and because it's a hereditary one, all they require is just whole blood. And then just a few others that are sort of rumbling around in the background I threw in on this one slide. It wasn't clear exactly 100% what they're doing, but they've got some panels going. They're looking at various types of diseases. They're using a little mixture of technologies. So you've got molecular diagnostic laboratories. They have a 20 SNP panel for the lung disease, which they call Respirigen. Emory Genetics, out of that's another sort of private-y, business-y type thing out of Emory University. Multiple non-cancer panels, so using next-gen sequencing. Insight Genetics are just looking at the ALK and KRAS as a companion diagnostic, and they're using qPCR. Tepnil's interesting because they're owned by Hologic, and they're kind of like a contract research org, but they use next-gen sequencing. I couldn't tell whether they're sort of a threat for you guys or whether they're just there to really work with Hologic to bring stuff in and move it out, and whether they're going to move up to something that could be of a competition. But Hologic, as you know, is a big company. They keep buying big companies. They bought GenProbe just recently for multiple billions of dollars, and that comes with a whole load of technologies and assays that go on their platforms. Illumina, they have a clinical services. Of course, naturally, they're using their HiSeq 2000, but that's really focused around whole genome sequencing. Totally different to what you folks are doing. But note that if Life Technologies are doing something like that, these guys are going to be in there too, especially with the GE Healthcare. And then the last one, which is more of a reference laboratory, is Arab. I think everybody here has heard of them. But they do a broad range of cancers, but they're only doing qPCR on fish. So again, be aware that there's these companies that do have all these cancers, that they do them. I've used Arab as well to have some sequencing done, or qPCR, sorry, done on some samples. And they're all over the place, as in the world. But it's not always NGS. So this is a real new technology. Okay, so you guys have a good, strong lead in saying we do next-gen sequencing, and there's speed behind getting the results and so forth. So, but there is competition, but as you see, nobody does it in the shortest turnaround time that you folks are touting. So that's good news. And that really concludes Module 4.